again. This is the last of our webinar series for this season. Antioxidants and redox medicine is so important for us to practice functional or integrative medicine or orthomolecular medicine, any way you want to call it. While our body, we have been endowed with endogenous antioxidants like glutathione, catalase, and SODs. But there are also antioxidants that are so important and vital for health, and unfortunately, we don't produce. That's one example is vitamin C. So today, Dr. Ron will take us through the role of antioxidants in oxidative stress and health. You were just sharing with me that, consider this as an introduction. Feel free to ask questions. There's so much more to learn, and we can always revisit this topic in future. Thank you very much, Ron. So I'll leave it to you now.
Thank you, Dr. SAHAMM. And once again, it is my pleasure to be speaking to all of you. This topic, I think I agree, it is one of the most important, but it's going to be somewhat new to most of you. It's new to me. And so Dr. Thomas Levy and I have been working together. And in the first part of my presentation, we have produced about a 7-slide summary of the basics, the fundamentals of redox medicine. And so these will be the ABCs that hopefully you will learn and begin to utilize. And then in the second part of my lecture, the major part of it, I will try to illustrate redox medicine as it applies to cancer. This is one of my lectures that I originally gave it in Minnesota. I have modified it, but I hope I can get some of these concepts across. Initially, you may feel like this does not have direct application. I'm not going to be talking about specific antioxidants or doses of antioxidants. It's more a conceptual presentation, but in that concept, you can begin to make decisions about how to approach your patients. It still contains all the basics of functional medicine. It's in many ways, it is the foundation of functional medicine. So I hope you enjoy the presentation today. I'm going to encourage you all. It's an expensive book, but it is a beautiful book written by Dr. A.S. Naidu called Redox Life. And this just kind of gives you an idea how much you can really dive deep into the emergence of redox medicine in our time. And so from this, I did take a kind of really high-level overview. And so I'm going to be talking to you this morning about redox and what I call the 5 phases of redox. So to narrow it down, to boil it down, all life is redox. And when there is stress or dysfunction or challenges, that's redox stress. But our cellular makeup is such that we are designed to meet the challenge of redox stress. But in order to create this, this kind of framework, I want to talk about the 5 phases of redox. So when everything is going well, we have good redox reserve. When stress or injury occurs, that is part of life. You cannot get through this world without experiencing some level of injury or stress. The cells have an amazing ability to let the system know that injury has occurred and the orchestration of repair is going to begin. And so the next phase of this is redox repair, and I will be defining exactly what that is. And then once everything has been fixed, then the cell will have established redox balance. So I'm going to take each of these concepts and now boil them down into principles. And so each one of these slides will be a series of principles. So if we look at redox life, basically redox in simple terms is the oxidation and reduction of biomolecules. Redox is continuous electron flow throughout the body. Life is flow, electron flow. We are electrical beings, and when everything is flowing, we're alive. But in the face of injury or toxicity, oxidation can begin to take over and deplete our electron reserves and flow. And so we depend upon reducing agents or reduction in order to restore this electron flow. So life, to boil it down, is the dynamic maintenance of redox balance. But without exception, we all in our trek in this planet, we will inevitably face redox stress. This is oxidative stress that occurs when there is pervasive electron depletion. The biomolecules become disrupted by electron depletion, and we have to restore the depleted electrons in order to restore proper molecular functioning. So unrelenting oxidative stress we can define as the root cause of all disease. I mean, these are big principles, but they nevertheless do hold true. So let's start out now with redox reserve. This is our general starting point. A reducing cellular milieu represents good redox reserve. We have to have reduction dominating. A predominance of reduced biomolecules assures normal electron flow and normal cellular microcurrents. As I stated, we are electrical creatures. That's why we can take our cell phone and we have an electrical field around our fingers, and we interact with the device because of the cellular microcurrents that we all have. That's how we think. That's how our heart beats. And so we are truly electrical beings composed of cellular microcurrents. Normal microcurrents support cellular health via normal transmembrane cellular potentials or voltage. Adequate redox reserves maintains cellular and organ health. We need this redox reserve in order to perpetuate these transmembrane cellular potentials. When there's redox injury, we're going to use a general term here, a toxin, and I'm going to use it in a very broad sense. In the course of my lecture, there are many things that can qualify as a toxin, not just arsenic or lead or some kind of snake venom. There are many more things that we'll generally define as a toxin, but it's any molecule or group of molecules that deplete electrons and cause redox injury. We're thinking at this point at the cellular level. Sustained redox injury leads to cellular dysfunction, and the location and the extent of redox injury defines the disease and its organ location. Just as a simple example, a heart attack. A heart attack is a major redox injury, but because it's happening in the myocardium, we define that disease as a myocardial infarction. But at the cellular level, it's a major oxidative event in the heart, simply because the blood supply is cut off. So multi-organ injury dysregulates life-sustaining function. So this is how we're starting to define chronic illness. So chronic illness, more often than not, involves multiple organs, whereas a heart attack obviously affects just one organ, but yet if we look at the roots of heart disease, we'll find that it's a multi-organ disease as well. Pervasive redox injury is the root cause of chronic illness. So this redox signaling, basically it happens once the toxic buildup starts to disrupt the redox reserve. The cells have to communicate with one another and within themselves. So dramatic oxidative events signal genetic expression. So this is the field of epigenetics. Once we experience something that's a dramatic injury or insult or toxicity to the body, that can signal genetic expression. Genetic expression mobilizes intracellular antioxidant enzymes and other adaptive molecules. The body's healing response to toxic injury is redox repair. And so redox repair is absolutely crucial to life. None of us can get through life without injury, so we have to have a good repair system. In a sense, the practice of medicine is the practice of optimizing redox repair. The primary redox repair molecule is ascorbate. And that may sound like, wait a minute, can we boil it down to just one molecule? Maybe yes, and of course, probably no. But in my lecture that follows, I'll try to make a pretty strong case that ascorbate may be, as Dr. Irwin Stone points out in his book, it may be the molecule that allowed life to evolve on this planet, which was not a friendly place. It's never been a totally friendly place. And so we need the ascorbate molecule because it has very interesting properties that I'm going to get to in my presentation today. Ascorbate is a molecular fuel for enhanced electron flow. It's an electron donor. Body-wide depletion of ascorbate is, of course, what we call scurvy. And scurvy, more often than not, results from pervasive toxic injury and oxidative stress. Of course, if your diet is poor or your intake of vitamin C is poor, that also contributes to scurvy. But more often than not, it's the depletion of scurvy due to pervasive toxic injury that is the main reason people get deeply into scurvy. Focal depletion of ascorbate is oftentimes organ specific. Obviously, if you have a depletion of scurvy in your skin, you'll start having bruises. Certainly, scurvy is associated with a number of illnesses. Your gums obviously become very inflamed if you're in a scurvy situation. So focal depletion is organ specific. Disseminated scurvy, though, results in multi-system dysregulation. And ascorbate in adequate amounts will neutralize oxidative stress and restore redox balance. Now, these are very high-level comments. They have to be interpreted in terms of the unique situation of each individual, but we're talking about fundamentals of redox. So once the body has restored electron flow, then you have achieved redox balance and cellular health. Cellular injury begins with excessive oxidative stress. Oxidative stress signals a process of redox repair, and scurvy results from inadequate ascorbate. Redox repair cannot proceed, but with adequate ascorbate, intracellular antioxidants can overcome redox injury. Restoring redox balance restores cellular health. Ubiquitous cellular health is the basis of the organism's maintenance of redox reserve and general health. So when it comes to functional medicine, we are basically trying to restore redox balance in our patients, and many of them suffer from greatly depleted reserves and huge amounts of oxidation within their body. So now I'm going to shift to the lecture. This was what I just finished, was more of a high altitude overview of redox medicine. And this lecture that I'm going to give now has been given in Minneapolis at an orthomolecular conference. And so Dr. Reardon was very close friends with Abram Hoffer. And when Dr. Reardon died, I was asked to become part of the Hoffer's board in the International Schizophrenia Foundation. And as a result, I took part in many of the orthomolecular medicine today meetings. And so I have a strong foundation in orthomolecular medicine. And so this lecture will have a kind of slant towards orthomolecular medicine, but even orthomolecular medicine is essentially, fundamentally redox medicine. Linus Pauling was definitely talking about redox medicine. I think he would be thrilled knowing that we are now really getting down to the molecular nuts and bolts of what constitutes true health. So orthos means, ortho means the right or the correct treatment, the right dose, the right way of doing things. I'm going to also define allopathic medicine is mostly just symptom suppression. It really doesn't get into the biochemistry of disease. It's more a classification of disease and pharmacologic interventions that are helpful with chronic symptoms. So to me, redox medicine, orthomolecular medicine is much more of a purer form of medical care. And if we can begin to understand it and become fluent in it, I think it will become the dominant way of thinking about how to work with chronic illness. So this particular lecture will focus on cancer. Sometimes I like to think of cancer as the ultimate chronic illness, and it certainly is one of the greatest challenges that we will all face. Now, this slide is a kind of table of contents of what I'm going to be talking about, and I'm going to be presenting these 14 concepts. And so this will kind of give you an idea of how I approach patients, and by all means, everyone's going to have a little bit different way of doing things. But this is Orthos as applied to oncology. So Orthos, generally speaking, is a repair process. And this yin and yang of redox life, redox stress, this is something we all face. And if it's not properly dealt with, one of the chronic diseases that can emerge, a dysfunction of cellular life, is obviously cancer. And so we want to use this knowledge of orthomolecular medicine and redox medicine to kind of give us an idea of how we approach patients. Now, Dr. Reardon, my mentor, he basically told people, we don't treat cancer, we treat the patient who has cancer. So he was looking at a more holistic perspective, which, you know, we're not just molecules, we're people. And so this personal dimension of care is just as important as our understanding of what's going on at the cellular level. I don't have time to go through all these pioneers. Some of you will recognize these very important names. Some of them I will mention in my lecture today, but we sit on the shoulders of giants, so I can't really take very much credit. I just feel very lucky that I happened to run into Dr. Reardon, and I've spent the last 31 years at the Reardon Clinic studying the principles of orthomolecular and now redox medicine, functional medicine. There's a lot of different names for what we do, but we are so, I think we're very fortunate. I think all of you that are listening are very fortunate that your mind is opened up to this understanding of health and disease and how we can work to maintain and improve our health and our longevity. So one of the terms that Dr. Reardon talked about, all of the great pioneers talked about is, and the whole essence of functional medicine is that we want to get down to root causes of the illness. And so redox medicine is the absolute ground zero of root causes. And so today I'm going to kind of give you a unified theory of causation, the personal and orthocellular processes that give rise to cancer. And as you can see from this slide, there are so many dimensions to cancer, and this is what makes caring for the cancer patient so complex. So you can just look through this and see that all of this can apply to cancer patients. And in some people, it'll be more one thing or another. So in very general terms, when an oncologist starts to address a cancer patient, he's really working to provide remedies for the disease. He determines the grade and stage. His treatments are an attempt to kill the cancer cells, but as a result, more oxidative stress is the result. And the yardstick by which they measure their success is the quantity of the patient's survival. The Orthos approach is more of an adjunctive care model. We're not trying to tell people that we're treating precisely their cancer in the same way that an oncologist does. We're looking at them as a whole person, epigenetically, providing adjunctive care for the whole patient, search for and correct many of the underlying causes that may be giving rise to the cancer, try to strengthen the healthy cells. I often tell my cancer patients that if you follow everything really correctly, you'll be healthier than you've ever been. That health may be the way you actually get over your cancer. So we're going to try to regulate oxidative stress, and our goal is to create quality of life processes in that patient's life. So the second major concept is that cancer is not a noun, it's cancering, it's a verb. As I'll show you, one of Dr. Reardon's kids, Neil Reardon, did an amazing job. He wrote a nice article on cancer as the non-healing wound. And so Hunninghake and Weinberg, in this molecular cancer research article, they have probably many of you have heard of this, the 7 hallmarks of cancer, the plausible mechanisms that give rise to cancer, self-sufficiency of growth signals, insensitivity to anti-growth signals. Here's that word signals. So signaling is, dysfunctional signaling is a major part of cancer. Evasion of apoptosis, unlimited proliferation potential. I'm going to show you a redox explanation for why these are some of the characteristics of cancer cells, the inflammatory microenvironment and altered cellular energetics. These guys are talking about redox medicine, and but that term is not utilized, but this today that's what I'm going to be trying to show you. Now this slide is another way at looking at cancer, and it's bringing in more of the personal dimension. Most of the people that develop cancer, they experience some kind of major stress in their life or chronic stress. Could be psycho-emotional, but nevertheless, it generates redox stress. I'm not going to go through all of these pathways. This is really a whole nother lecture, but some of what I'm going to say, I'll come back to this slide. But the net result of all this is cancering, the process of cells changing in a low oxygen environment and becoming cancerous. They start to function as if they were trying to survive in a very hostile environment. So we'll have more to say about this in later slides. So here's Neil Reardon, Dr. Reardon's son, that did a lot of the breakthrough research of RECNAC, the organization that Dr. Reardon founded to understand how IV vitamin C can help cancer patients. But what Neil pointed out is that cancer is a non-healing wound, mostly due to the fact that chronic injury persists, and it can be physical, chemical, biological, or even emotional or mind-body injury. And all this injury creates a wound that never does get the right resources to heal. I want you to think of all these items that are listed in this particular slide as toxins. Now, obviously, you don't think of radiation as a toxin per se. Toxic chemicals are toxins. Infections, though, if you talk to Dr. Tom Levy, infections create toxins. And in the field of emotions, I'm sure all of you know that toxic emotions are very real and they can create disease. And so we're looking at a whole person perspective, but cancer is in general terms a non-healing wound. Now, coming back to this concept of signaling, the RECNAC team utilized a cytokine array looking at all of these signals, cancer growth signals. And these signals represent a kind of fingerprint that can show us what's going on in terms of apoptosis, inflammation, angiogenesis, atherosclerosis, all of the various ways that the body will try to respond to a chronic illness. And so what they found was that when you look at cancer cells, the various cytokine signaling is an attempt to heal a wound. Cancer is a wound that for whatever reason can't heal, and so it continues to grow. What the RECNAC team was looking for was ways to turn off this overabundance, overproduction of wound healing cytokines. And so it does turn out that IV vitamin C, even 1 gram vitamin C will cause a dramatic shift in cytokine activity back towards more normal. So this is an example of giving ascorbate, which is a major electron donor, as a way of toning down the signaling that drives the abnormal cancer activity. So they found that 22 categories of cytokines were improved with a whole series of IV vitamin C. And that is one of the ways that IV vitamin C is beneficial to cancer patients. This is just more of that research showing vitamin C reduces that cytokine production, at least in vitro. So we have good evidence that vitamin C is helping to change that signaling that drives the cancerous process. So this is where I'm going to show you a redox model of wound healing. We could say that all disease is a kind of wound. Obviously, it's not your typical skin wound, but there is a wounding that occurs in cells. And so this will represent what I conceived as a redox model. And I can only give a lot of credit once again to Dr. Naidoo's book. Basically, the essence of the book is that life is electron flow. And here's a picture of Dr. Irwin Stone, who wrote The Healing Factor. This book is available online. I encourage all of you to download it, find it. I think it's on the Vitamin C Foundation website. It's a wonderful book showing that really life could not have made it on the planet without probably ascorbate as the ultimate electron donor. So here's his quote: "On a molecular basis, the whole living process is nothing more than an orderly flow and transfer of electrons." So this will be my simple redox model of wound healing. And so when we're happy and healthy, we are adapting to our environment, but inevitably injury does occur, and that results in signaling, and the signaling starts a process of repair that, if all goes well, we will be back to a state of homeostasis. The mitochondria, the electron transport chain, plays a major role in this healing process. So now to put in the different elements, redox balance is where we're adapting. Injury is simply that disruption of redox balance. Oxidation is itself a signal, and that signals the coordinated repair process that we call inflammation. Inflammation is not our enemy, it's our friend. But what happens in most chronic illness is the inflammatory process becomes dysregulated. And healing is nothing other than redox life processes, the dance, the coordinated repair process that the body miraculously is able to provide. So in real simple terms, redox stress is chronic injury. This is the fourth concept that I want to get across to you. And there is no lack of external oxidative stress that all of us must deal with in our lives. I'm not going to cover every one of these areas, but under the category of lifestyle, social, environment, and miscellaneous, there's all kinds of things that create external oxidative stress. And that's the way most of us think of stress, is that it's an external affair. But what we find is that there is such a thing as internal oxidative stress at the molecular level, within the mitochondria. Here's the matrix of the mitochondria, and you can have fuel and energy homeostasis imbalance, fatty acid metabolism abnormalities, oxidative phosphorylation impairment, mitochondrial gene expression damage and alterations, inflammatory response impairment, increase in ROS, reactive oxygen species, and a decrease in ATP. Cancer patients are all tired, and they're tired because at the molecular level, they are not generating enough injury. And what we see happening in our world is this massive, massive rise in obesity and diabetes and insulin resistance. And so all of these cellular responses very slowly create these chronic illnesses that are associated with insulin resistance. So insulin resistance can be a big factor in all of this. So this is a pretty complicated slide. I'm going to break it down in future slides, but this kind of gives you a panorama view of what happens in chronic illness, that external and internal stress triggers injury, which triggers reactive oxygen species and nitrotive oxygen species, but the signaling is dysfunctional, the wound doesn't heal, and so we get stuck in this loop. So it's unrelenting redox stress that creates dysfunctional cellular healing. And I'm going to say this is the true root cause of all chronic illness. And we'll kind of define that in more clinical terms here as I get further into the presentation. So dysfunctional cellular signaling is probably the true cause of cancer. So there's a kind of yin and yang that you need to be aware of, you know, that reactive oxygen species, this is oxidative stress, but these same oxidative species act as signaling oxygen species. So that's why I sometimes refer to them as SOS. SOS is in green, signaling oxygen species. These are not the bad guys. This is how the cells coordinate the healing response when there is injury. So, and oxidation obviously is not always bad. We need it for good immune system and for our cells to make hydrogen peroxide within lysosomes in order to attack and destroy bacteria and viruses. We need it for energy metabolism. So there is good cellular oxidation. So I'm going to call that SOS, signaling oxidative stress. But of course, we talk about bad cellular oxidation, and we know that inflammation and aging all arise out of oxidative stress. And look at all the various chronic illnesses that arise from that. So the true fundamental cause of most of the chronic illness in the modern world today is related to inflammation and oxidative stress out of control. And so that creates the reactive oxygen species, the free radicals that set up chain reactions and damage cell membranes, mitochondria, DNA, the degenerative diseases, cataracts, macular degeneration, cancer. All of these are the results of imbalanced cellular oxidation. So health is where we have developed this redox reserve. It's a healing cellular environment where we're using oxygen optimally. We have a strong NAD to NADH ratio. This is going on in the matrix of the mitochondria. There's abundant ATP and energy. We take in adequate water and nutrients, antioxidant enzymes, and we have healthy lifestyle choices. This is health from a redox medicine perspective. What I'm going to show you is that when things go south and we start to get sick, then that milieu shifts to a more oxidizing cellular milieu with stress and depletion, accelerated aging. And I'm going to highlight in this presentation poor oxygen utilization. That depletes NAD, that lowers our ATP. It allows metals and toxins to build up in the cells. It's supported by poor food and malnutrition and unhealthy lifestyle choices. Now, over here is signaling oxidative stress or oxidative oxygen species, signaling oxidative species. Here is reactive oxygen species. These are both oxygen species, but depending upon the balance, they can turn from being a good thing to being a bad thing. And so this poor oxygen utilization, this is something that really has been an eye-opener for me. This triggers something called the HIF-1 alpha. It's the hypoxia induction factor. This is what is the beginning of all chronic illness, and it plays heavily in cancer. And interestingly enough, ascorbate will attenuate the hypoxia induction factor. So now we're going to look at redox reserve. It's a reducing cellular milieu. It represents good redox reserve. A predominance of reduced biomolecule assures normal electron and normal cellular microcurrent. So basically what I'm doing here is repeating the principles of redox medicine. Normal microcurrents support cellular health via normal transmembrane potentials or voltage. Adequate redox reserve maintains cellular health and organ health. So these are the signaling oxidative species, oxygen species. So that's redox reserve. So then moving over to redox injury, it's any toxin, any molecule or group of molecules that deplete electrons and cause redox energy. And these toxins can arise out of poor lifestyle choices as well. This creates cellular dysfunction. The location and extent of the redox injury defines the disease and its organ location. Multi-organ injury dysregulates life-sustaining functions. Most of your chronic illnesses are not organ-specific. They are dysfunctions of all the various orchestration of health. And so we could call disease bad music. The body's out of tune, and it can kill us. So it's very important. So this is a wounded cellular environment. This is redox injury. It's basically a wounded cellular environment. It's reactive oxygen species is the highlight of this, and you hear about ROS all the time. So I've been moving through my 14 concepts. I'm now going to get down to concept number 6, which is dysfunctional oxygen utilization. This is very, very important concept to understand. And so if we look at this model of chronic illness, here is under structure and function. Structure, we have DNA and membrane. Function, we have cellular respiration. And if that is out of balance, then we're tilting towards more chronic illness. This is how illness starts. So I'm going to fill in the model here. Here's external oxidative stress and internal oxidative stress. They kind of overlap with one another. This is what results in injury to the cellular level. And then we start having structure dysfunction. We have altered functioning of the cellular respiration. That disrupts cellular signaling, and the signaling dysfunction gives rise to more reactive oxygen species, and we have the non-healing wound. And this is true pathos or pathology. This is I guess you would say, this is what I would refer to as redox pathology. And it is the fundamental cause of disease. Now, out of all of this that I just showed you, dysfunctional oxygen utilization is probably the key thing to understand if we're going to understand how we heal redox injury. Now, I have to give credit to my mentor here, Dr. Frank Schallenberger. He has a wonderful book that I'll show you in just a minute, but he has really gone into the measuring and assessing mitochondrial function in his patients, and he uses this analyzer, this bioenergy testing, in order to define the energy quotient of his patients. And basically what the energy quotient refers to is the efficiency that oxygen is being utilized at the mitochondrial level. And Dr. Schallenberger has found that there's a number of things that create poor oxygen utilization. And this is his chart. And for those of you that saw my first lecture several weeks back, you'll see that these are how, at the clinical level, people get into early onset mitochondrial dysfunction, poor oxygen utilization. These are the root causes: deficient nutrients, poor thyroid function, poor fat utilization, not sleeping well, increased stress, deficient hormones, excessive chronic inflammation, toxins, infections, ischemia, low NAD to NADH ratio, poor methylation, not enough movement, and insulin resistance. This is all carefully outlined in his wonderful book called The Principles and Applications of Ozone Therapy. There are 3 chapters in this book that are fundamental to the understanding of redox medicine. These are the factors that decrease oxygen utilization, and that can occur in apparently healthy people. He has found, when he did studies on populations of even young people, 20s and 30s, that they had the signs of early oxygen dysfunction. And these are the same things that create reactive oxygen species. Basically, what's in this upside down pyramid are the same things that create dysfunctional oxygen utilization. And as a result, what happens when this occurs is you get the HIF-1 alpha transcription factor induced. It's induced by stress. And so this is a fundamental cause of dysfunction within the body. Now, what I'm going to show you next is probably controversial. It's not been proven, but your blood level of ascorbic acid may be an important signal to your body in terms of whether or not it should start shifting its functioning. In other words, if you have inadequate amounts of vitamin C, that may be one of the core reasons why your body is shifting towards hypoxia. All of these factors tend to be all of these factors over here tend to be activated by poor oxygenation. And so when that happens, you get this HIF transcription factor creates all the play of these abnormal cytokines. You start moving into more of a cancer type of physiology, angiogenesis, cell proliferation and survival, metabolic adaptation, glycolysis, and all of this promotes tumor growth, invasion, and metastasis. So cancer is really the outcome of long-term imbalance in your utilization of oxygen. So it is truly a redox disease. This really struck me years ago, and this was a paper that I was writing down the chemistry of HIF, and then I was surprised to find out that vitamin C attenuates HIF. Vitamin C attenuates hypoxia induction factor. And so here's a couple slides that show that as you increase the amount of ascorbate in a tumor model, the HIF goes down. And here is just one in this on the right hand side. Here is just one infusion of high dose vitamin C. You can see the drop in HIF-1 alpha, But if you just do one infusion and don't perpetuate your vitamin C, it will come back. You know, one vitamin C infusion or one dose of vitamin C is not the answer. It's a continuous infusion that's going to be an important part of controlling HIF. So we know that the chronic wound and the vicious cycle of cellular hypoxia, that's due to the fact that you're not oxidizing very well. You're not forming what Dr. Schallenberger says, an adequate amount of NAD. That slows the Krebs cycle and you do not make ATP. And if you do not make ATP, you cannot make your antioxidant enzymes like glutathione and dismutase properly, catalase. And the result is increased mitochondrial injury. So this is a vicious cycle, and this induces anaerobic functioning within the body. And one of the results of that is p53 dysfunction. The p53 gene is the gene that normally would take out the injured cell. Now, this is the novel idea. Could vitamin C be an intracellular distress signaling system? That is the lack of vitamin C. And so one of the questions that I've always entertained is why, you know, vitamin C is so important. Why did human beings lose the genetic ability to make our own vitamin C? What was the survival advantage of not being able to make our own vitamin C? And it turns out that that vitamin C is a kind of link to our environment. Our early ancestors, if they had access to high vitamin C level foods, they tended to survive better. So what this author is saying is that it's epigenetics. Dietary vitamin C is episodic and unpredictable. It's when it's not available that you tend to get into trouble. So it's the unavailability at certain critical times in the animal's life that's important. So in other words, the survival of that animal depended on access to vitamin C. So it was a kind of survival of the fittest. So this was the advantage. If we had good vitamin C intake, we had a better likelihood of survival. And that's even more so true in the modern world because we have a hugely toxic environment. And for those people that are not eating well or not taking advantage of our knowledge of vitamin C, their chances of survival are going way down. And that is becoming very evident in the pandemic that we're currently in. People who have adequate nutrient reserves, have adequate vitamin C, good nutrition, you know, vitamin D, these are things that are allowing them to survive. The people who are sickly, diabetic, low nutrient levels, they're the ones that are just not making it in the ICUs. And so it turns out that this ability of vitamin C to neutralize the hypoxia induction factor is extremely important. Now, this is where I'm probably going to lose some of you, but I think it's, and I apologize if I do not express this as clearly as I would like to, but I'm going to talk to you about the ascorbyl radical at this point. What is it about vitamin C that makes it so darn important? And it's the fact that vitamin C is a kind of antidote or a kind of a balancing factor to the reactive oxygen species that get triggered by all these various things that create stress within the body. These same things that trigger the superoxide radical are the same things that trigger the ascorbate radical as well. And the ascorbate radical is the counterbalancing element to this oxygen radical. So let's just take a look here real quick at how the molecular structure of vitamin C. You know, we could spend like 20 minutes on this one slide, but I'm not going to do it. You know, when we take ascorbic acid and put it into water, there's a pK constant and it dissociates and goes into solution. It's an acid. And then when it gives off an electron, one of its 2 electrons, it becomes what's called the anion form, the ascorbyl radical. And so there's some very interesting fact. Now, then if that loses the 2nd electron, then it becomes dehydroascorbate. Then it's basically spent. But there's something very unique about this ascorbyl radical that we really need to know about. And this was fascinating to me, that the ascorbyl radical is a stable free radical. That goes against what probably most of us learned in chemistry, is that free radicals are very unstable. They want to quench that free electron, and so they will steal electrons. But the ascorbyl radical forms this what's called a pi system, that this 1 electron gets it's a very interesting term. It gets domesticated over these 6 atoms. It's kind of forms a little cloud, and it's pretty stable. And it itself does not make a superoxide. In other words, it doesn't create more free radical damage. It's basically a kind of a it's kind of a sentinel. Here it is. It's very unreactive, and it forms this little cloud. And so it's kind of like waiting. It can either give an electron or it can receive an electron. So Dr. Levy had a very interesting way of explaining this. He said, because of its stable delocalized electron, the ascorbyl radical can accept or donate an electron. It forms an electron buffer. This is why it's so important. It can donate an electron and become dehydroascorbate, or it can receive an electron from, for example, the superoxide, and it can help neutralize the superoxide. But it itself does not create superoxides. So it serves a very interesting stabilizing function within the cell. Now, this was ascertained back in 1976 by Dr. Irwin Stone, and I normally don't read slides, but I think this one is worth reading. Chemically, ascorbic acid is rather simple carbohydrate related to the blood sugar glucose. Unlike glucose, it contains an unusual reactive combination of molecules called the indodiol, or the pi system. The presence of this group confers upon ascorbic acid molecule certain unique biochemical characteristics which may explain its vital importance in the living process. It transforms a relatively inactive sugar into a highly it's not as highly reactive as he says here, but it's a reversible carbohydrate derivative that donates or accepts electrons from its surrounding medium. This is technically known as an oxidation reduction system. So you could see here that Dr. Stone was talking about redox. It's a redox system. And one of the interesting things about a redox molecule is it can give or it can take. It's a bi-oxidant. So the bi-oxidant properties of vitamin C, especially intravenous vitamin C, is that it can act as an antioxidant, mops up free radicals, modulates cytokines, promotes the formation of collagen to wall off tumors. But at the same time, it can also work as a pro-oxidant. And this is where it can donate an electron to iron, and you create the reduced form of iron, which interacts with oxygen to create the superoxide. And then the superoxide interacts with water to create hydrogen peroxide. And so that's our mechanism for how high-dose vitamin C works against cancer and stimulates apoptosis. So I don't know if all of you followed that, but to me, it is profound stuff. What Dr. Reardon did is he showed in cell culture that if you give an adequate dose of vitamin C in the cell culture, there was decreased survival of the cancer cells. And he looked at 60 different types of cancer cells. He grew them in culture media, and he found that somewhere between 350 to 400, it would knock out just about all of the cancer cells. So this is the basis of the Reardon protocol, where we recommend people to work up to an adequate dose that generates a post-IVC saturation level of about 350 to 400. This was all done in vitro. Now, this is another experiment that he did that I think is even more interesting. What he did is he took a prostate cancer patient and drew blood from him to create a blood culture. And what he found is that when he grew prostate cancer cells on this gentleman's blood culture, they grew fine. They grew, their percent survival was 100%. But then what he did during the infusion of a 60-gram IV vitamin C, he withdrew that blood from this cancer individual and used that blood for a culture media, and the cancer cells would not grow. And so when the blood level was above the 350 to 400 or the 20 millimole level, there was essentially no growth of cancer cells. So this is a verification of the Reardon approach of using the high level of IV vitamin C. So this was not published as much, but I think it's a neat experiment. I won't have time to go into this slide. Basically, what we're doing at Reardon Clinic is looking at all the different dosage ranges. And I will tell you that Dr. Hickey's idea of dynamic flow, we shouldn't forget about that. Right now, we're looking at the effect of continuous IV vitamin C at the 1 to 2 millimole level, as well as combining different elements of high-dose vitamin C, mid-dose vitamin C, and dynamic flow oral dosing in order to get better results with our cancer patients. So now there's a big advantage of giving IV vitamin C, and that is it is a non-rate limited advantage. So in other words, vitamin C can just be an electron donor, but you don't have to, if you're giving it in large doses, you don't have to revitalize the molecule. It's what was called a non-rate limited antioxidant free radical scavenger. So you can use vitamin C, it can donate its electron, and it can take glutathione and recharge it. You can take the oxidized glutathione and recharge it. And so this is why vitamin C is very important in terms of maintaining intracellular antioxidant properties. The cell itself can regenerate glutathione, and as you know, glutathione is a critical molecule for resisting oxidative stress. Very, very important. But in order for glutathione to work, you need to have adequate selenium, magnesium, zinc, vitamin C, the B vitamins, folate, cysteine. In other words, you have to have your methylation pathways working. And if you don't, or if you use up these nutrients in a really high stress, high oxidative stress situation, the GSR is not adequate. So, so this is why giving large doses of vitamin C, it's a rescue molecule, and it's non-rate limited. As long as you keep giving it, Dr. Robert Cathcart found that in patients that were severely sick with mono, for instance, he could give them up to 200 grams of oral ascorbate without causing diarrhea, because he was fulfilling that need for ascorbate to put out the fire of oxidative stress. And so the other thing that vitamin C does is, this could be a whole nother lecture, is one of the causes of aging is what they call electron leak, or reactive oxygen species leakage. And so ascorbate, there are SVCT transporters that can get ascorbate into the matrix of the mitochondria. So don't underestimate the power of vitamin C. It's just that it has to be dosed in a pretty aggressive way. So now I'm going to come back to this hypoxic intracellular environment idea that basically cancer cells are growing in an area of cellular hypoxia. And that's due to the microcellular environment of a chronically injured area of the body, such as smoking injures the lungs. You know, asbestos can also do the same thing. And these are chronic injuries that create cellular hypoxia and inflammation. And that environment promotes tumor progression and tumor invasion and metastasis. And so I wanted to just show you, and this is, all of you are aware of Otto Warburg, that if you take and put any healthy cell in an anaerobic environment, it will start to shift towards cancer. He demonstrated that all cancer cells have become anaerobic obligates. And so what I thought I would do is kind of take you through this step by step, just to show you redox medicine kind of like in action. And so this is this slide will be a dynamic summary of everything that I've been talking about. And I realize I've been conveying some difficult concepts, but kind of follow me on this slide, and I think it'll put it all together in your mind. And so again, in Dr. Schallenberger's book, he basically says if you cannot oxidize your NADH to NAD, then you're going to have a low ratio of NAD to NADH, and that will result in poor electron transport. So this gets into the electron transport chain and the dynamics of that, which we don't have time to go into great detail right now. But the net result of that is that the Krebs cycle starts to slow down and you get decreasing ATP. This is why all cancer patients are tired. And this is why chronic illness almost always involves fatigue, because we have a decreasing production of ATP. And in cancer, when the cancer cells shift to glycolysis, one molecule of sugar is only generating 2 ATPs, whereas if you can run it through the mitochondria, you can generate 36 ATPs. So what's happening is in disease, the body is shifting more to glycolysis, which is it's only doing that because there's a lack of oxygen. And so as a result, because you do not have enough ATP, you have a reduced production of antioxidant enzymes. You cannot make enough glutathione reductase, and so you cannot quench the electron leakage, and so you start having more and more damage to the mitochondrial DNA and the mitochondrial membrane, and the result is you have severe mitochondrial oxidative stress. Your mitochondria start working properly, and without mitochondria, your P53 gene becomes dysfunctional and apoptosis cannot occur, and so cancer cells start to shift more and more anaerobic. So anaerobic functioning is the essence of cancer. And when the cells are functioning anaerobically, they are not generating carbon dioxide, that you have decreased carbon dioxide output. And so the result of that is you need carbon dioxide to displace oxygen from the hemoglobin. And so there's this vicious cycle. The worse this whole situation becomes, the less likely you're able to get oxygen to the chronically ill tissue. And so this is why cancer is a kind of a progressive process, and cells get locked in chronic hypoxia. So there's your hypoxia, and that induces the HIF transcription factor based upon this cellular hypoxia, which is part of a vicious cycle. This is redox disease right here. And the net result of that is progressive oxidative stress. And so you have reactive oxygen species galore. So, and you lack your good old, if you don't have enough vitamin C, you can't convert this, you can't heal it. So this concept is not new. If you want, you can go to this web address in orthomolecular.org, and you have access to the Journal of Orthomolecular Medicine. And this whole concept was very, very well explained by Stephen Levine and Paris Kidd in this book. It's an article called Beyond Antioxidant Adaptation. And basically they're saying what I just said, that hypoxia is the key factor in degenerative disease, especially cancer. And you get oxidative damage to DNA, you get all kinds of kind of progressive degeneration, and cells get locked into this. And this is why it's so hard to reverse cancer once it's built up speed, so to speak. So as you have increasing levels of oxidative stress, that stimulates more cell division. There is some apoptosis and necrosis that will occur, but that creates more inflammation. So these factors drive oxidation and create more and more cancerous cells. So what if we, that's one of the problems of trying to kill cancer. When you kill cancer cells, you create more oxidation, and that drives more of the oxidative stress, and you're back into the vicious cycle. And this is why chemotherapy and surgery, more often than not, only buys the patient time, but all of that oxidation promotes the cancerous process rather than healing the cancerous process. And so let's go back to the model here. And would there be a different way? Could we use an orthos approach to cancer? What would we be doing different in that particular approach? So here are all the oxygen species. I'm not going to go into these. Dr. Levy is getting really interested in hydrogen peroxide, which can be formed from the superoxide radical. And he's finding that it is a very important part of the healing process. And so hydrogen peroxide and vitamin C kind of work together. So in general, what we have to do is we've got to reduce the reactive oxygen species. That's basically what it boils down to. And so we do that by encouraging our patients to take better care of themselves. I mean, after all that heavy-duty discussion of redox medicine, in many ways, it doesn't change the practice of functional medicine. We want our patients to have adequate nutrients. I'm really big on thyroid. As you remember from my lecture on reverse T3, you've got to have proper thyroid functioning. That's very important up here. You've got to try to reduce the breakdown of fat, improve people's sleep, reduce their stress, address causes of inflammation, use anti-inflammatory strategies, correct their hormone imbalances. Dr. Schellenberger, of course, uses a lot of ozone therapy. That would help them get rid of chronic infections, dental infections, infected teeth, infected tonsils. These are things that Dr. Levy's very interested in. Basically detoxifying, improving methylation, get regular exercise, and reduce insulin resistance. And this will improve circulation through exercise and whatnot. So you can see by dealing with root causes, all of these things will improve oxygen utilization. So in this case, we're not just killing the cancer, which is what the allopathic remedy is to attack the cancer cell, the tumor cell. Here, what we want to do is we want to reduce all the factors that are causing reactive oxygen species in general in the body. And so this is an orthos approach to dealing with cancer. Care for the whole patient, search for and correct underlying causes, strengthen the healthy cells, regulate oxidative stress, improve quality of life processes. So, so you've got to basically regulate internal and external oxidative stress. And so we're back to this diagram. A lot of these factors are basically redox stressors that create cortisol, which depletes adrenaline, That depletes vitamin C. You know, vitamin C is involved in the process of making adrenaline. Adrenaline kind of breaks down, you need adrenaline to stimulate oxidative phosphorylation. And when that stops working, the body shifts to anaerobic glycolysis, lactic acidosis. That creates an environment that's favorable to fungal overgrowth, yeast overgrowth, and that can result in DNA mutations. Over here, when you're under a lot of stress and you don't sleep very well, you will deplete your melatonin dramatically. It also depletes IL-2, which results in immune suppression. You also have a depletion of dopamine and tryptophan, and you cannot synthesize niacin. You know, niacin was a very important part of Dr. Hoffer's treatment of cancer patients. He used high-dose vitamin C and niacin or niacinamide along with some other key nutrients, and was able to get marked increased survival time in his cancer patients, even without IV vitamin C. So in general, we want to reduce oxidative stress. We use vitamin C. This can help restore mitochondrial functioning. So this is the way we systematically deal with the non-healing wounds of cancer. And so I've kind of shown this as a green slide. We want to promote more of the signaling oxygen species through better sleep, increased movements, nutrients, light, adequate hydration, care for the brain, care for the gut, care for the liver, lungs, heart, and thymus. We want to get our thymus glands working so we get those lymphocytes back in there, helping us against the cancer. So all of these are what we call wise choices as opposed to poor choices. So there was a famous doctor, Jonas Salk, who discovered the Salk vaccine. He wrote a beautiful little book. He actually lectured at our medical school. The book was called The Survival of the Wisest. Life is an error-making and error-correcting process, and nature in marking man's papers will grade him for wisdom as measured both by survival and by the quality of life of those who survive. So even though we're talking about life at the molecular level, our everyday choices have a big impact on what happens at the nanoparticle level. And so this is my slide basically saying that if you want to heal cancer, your life needs to be process-driven by this concept of orthos, better self-care, co-learning. And these are some of the biggies. You can look at this slide. These are the things. You got to get your adrenals working. You got to get your thyroid working. You got to get your various organ systems in tip-top shape. I put vitamin C. The vitamin C radical, the stable radical is really the essence of a healthy life. So so never forget vitamin C. Of course, vitamin D is a foundation. Niacin is highly important. Methylation, breathing, oxygen utilization. All of these things go into orthos, the balancing of life processes. I'm going to finish my presentation here with just a brief discussion of melatonin. As I pointed out here, one of the factors that happens with chronic stress is a depletion of melatonin. And melatonin, we now know, is made in large amounts within the matrix of the mitochondria. Melatonin is made at night, and we can measure that in the blood, but every cell in the body that has mitochondria is using melatonin as a redox quenching molecule. IVC, vitamin C, can quench 2 molecules, but we now know that melatonin, a melatonin molecule, can quench up to 10 free radicals. It's pretty amazing. And I'm going to have you, I'm running out of time here. I want to leave some time for questions, but this is a discussion of how melatonin moves easily into the mitochondrial matrix and does a lot of good in terms of reducing oxidative stress and regulating those rogue electrons that can occur in the mitochondria. So this I'm going to have to save this. This is a very complex slide, but it looks at the fact that if you've got nitric oxide along with superoxide, these create something called the perioxide nitrate, which is an even more horrible free radical that is doing all kinds of damage in modern times. High levels of oxidative stress, nitrogen stress, mitochondrial dysfunction, autonomic dysfunction, and cytokine storms that trigger chronic inflammation. Martin Pall has done a lot with this vicious cycle of the oh no vicious cycle. And what it does is it causes more calcium to go into the cells. And as Dr. Levy will tell you, that calcium in the cell can cause cell death. Intracellular calcium buildup is lethal. And so this would get into a discussion of these voltage-gated calcium channels inside the cell, that if you have a lot of oxidative stress, this causes redox chaos in the cells. And this leads into a whole discussion about Wi-Fi and microwaves and that whole thing, but I just don't have time to get into it. But melatonin is a really good antidote. As we age, I'm getting up there myself in these higher age groups, we don't make very much melatonin. And so you can take melatonin during the day and it will not make you sleepy. And so we're finding that you can use these really high doses of melatonin very effectively against cancer. And I won't have time to go into this in great detail. If you go to YouTube, Reardon Clinic channel, if you scroll down, Dr. Schallenberger did a beautiful lecture on melatonin and cancer research. And so melatonin can be a very effective way to slow cancer using it at high dosages. And Dr. Russell Ryder, who has for the last 45 years has published over 250 studies on high dose melatonin. It's been totally overlooked as a very powerful redox molecule. So don't forget melatonin. Dr. Schallenberger uses 60 milligrams 3 to 6 times a day in his stage 4 patients with great effectiveness in dealing with advanced cancer. And so this can be a very efficient way to add on. It's like an add-on to vitamin C. And so when we look at Hanahan's and Weinberg's cancer hallmarks, it turns out that melatonin does a very good job of blocking cancer signaling, stimulating intracellular glutathione, modulating the NF-kappa B induction, reducing aneuploidy, and inhibiting VEGF production, increasing P53 expression, inhibiting the hypoxia induction factor, and strengthening these cadherin junctions. So I can't take time to explain all that, but it's a very powerful cancer therapy. So we need redox medicine for a simple reason, that chronic disease is growing exponentially. In the last 25 years, in the last generation, there has been a dramatic growth in autoimmune diseases, neurological diseases, metabolic and inflammatory diseases. So 40 of these disorders have doubled in just the past 25 years. And plus there's a bunch of new ones. And so take a look at this slide. This comes from a very reputable article showing how these numbers have increased beyond comprehension in our world. And these are the people that you're seeing. And this all goes back to redox dysfunction, huge redox dysfunction in our world right now. And yet we're trying to solve it with medications that treat symptoms. Until people get to the true root cause of chronic illness, we won't be able to solve these huge chronic illnesses, including the pandemic that's currently afflicting the world. There are a number of pandemics in the world right now. The virus is just one of many. And so I hope this presentation has gotten you interested in redox medicine. Put another plug in for this wonderful book by Dr. Naidoo. It's a great book to spend some time with and just see how pertinent this discussion really is to the practice of medicine. So for those of you that have listened in, I think this is on the cutting edge of medicine today, and hopefully it will inspire you to learn more about how to use redox medicine more effectively in your practice. So thank you very much.
Thank you very much, Ron. We'll take questions now.
Dr. Maya, you have a question?
Yeah, good morning, Ron. Good morning
Yes.
at this time. Okay, I have actually two questions. The first one is you mentioning a lot. You mentioned, it's very interesting, the lecture, and you mentioned a lot from thyroid to melatonin to dopamine, tryptophan, niacin, vitamin C, everything. But how about can we actually take the mitochondrial antioxidant like CoQ10, PQQ, MyToQ to our arsenal to help, to help for the better oxygenation?
Sure.
First question.
Sure. There was no way I could mention all the various nutrients that affect mitochondrial functioning, but PQQ, CoQ10, a lot of these are methylation factors. And so one of the kind of the more fundamental ways of dealing with the mitochondria, interestingly enough, is to help improve methylation in your patients. And so take a look at their homocysteine, take a look at their B12. We look at glutathione levels. Your body needs methylation in order to generate adequate amounts of glutathione. So methylation is indirectly a redox medicine approach.
Okay, and thank you. And the second question is, I'm rather, I'm always concerned if patients or myself take a lot of melatonin at night, since we are not sure about our cortisol. Is it so that I have to take cortisol in the morning? I mean hydrocortisone if I take too much melatonin at night, since feeling kind of dizzy, or please say something about that.
Well, I really was only introduced to the melatonin story about 3 years ago at a Dr. Schellenberger conference, and I happened to sit next to Dr. Russell Ryder, who is in his mid-80s, and he's been taking 180 milligrams a night of melatonin for the last, I think he said, 15 years. The guy did not look 86. He looked like he was in his 60s. So it has an anti-aging effect. He has studied it in mice. He's given mice as much as 300 milligrams daily for 2 years with no adverse effects to their endocrine system. There are a number of people that also look at melatonin as a way of, it's another alternative way of helping women with menopause. It also is very effective at preventing Alzheimer's. And in my own experience, since working with and getting to know Dr. Ryder, I've been taking 100 milligrams of melatonin at bedtime for the last 2 years, and I have not experienced any drop in cortisol. So if that is a concern, I can assure you it has not been anything that I've observed. Now, what I have observed is when people first start using melatonin, because it is such a powerful antioxidant, some patients will have a Herxheimer type response and they won't sleep as well. So I always start patients at very low amounts, maybe 3 milligrams, 6 milligrams, 10 milligrams, and just very slowly work their dose up so that they don't have a disruption in their sleep. But once they are stable at any level, you can go up to a higher level. And when you get into the 20s and 40s, you can go up faster without causing any disruption in sleep.
Thank you.
You're welcome.
Anyone else for questions? Please unmute yourself.
Yes, this is Chan Kudinak. I was educated that melatonin, if you take too much external melatonin, you can suppress your body's natural ability to
Hello, Chan?
Hello.
melatonin. And I was also taught that your
Chan,
mitochondria
you need to
produces it.
repeat your question, Chan.
Okay, I'm going to type the
Yeah.
question. Yes, melatonin, if we take excess, hello, if we take excess amounts, I was told that your body's that true.
I wanted to share the slides again, but they came off. We only make melatonin in large amounts in our youth. Babies make very little, but about 2 or 3 months into their life, their level will go from 10 picograms per milliliter up to 130 picograms per milliliter. Then when we reach puberty, our melatonin dramatically drops. And by the age of 50, 55, we're back down to less than 10 picograms per milliliter. So it is possible, if you were dealing with someone who is in their 20s and 30s and you use the higher doses of melatonin, you would suppress some of their melatonin production. But it's kind of like the same way with other hormones. If you give enough of the hormone, it becomes a hormone replacement therapy for people. So think of melatonin, especially for people over the age of 50, as hormone replacement therapy on a dramatic scale.
Thank you.
Anyone else, please? Any questions?
Hello, Dr. Ron, can you hear me?
Yes, I can.
My name is Dr. Baljit. Do we have any sort of evidence or data about how high melatonin production might have been in ancient man a long time ago?
No, that's a very interesting question. I don't know. Dr. Russell Reiter, if you type in Reiter, R-E-I-T-E-R, in a search engine, that man has done hundreds and hundreds of studies on melatonin, and it has been blatantly overlooked as a very powerful therapeutic tool in terms of a redox regulating molecule. And so I would say, and you might even call him, he's at the University of Texas in San Antonio. He's very approachable. He's a really nice guy, and he's written a book on melatonin, but I don't know the answer, but I'll bet you he would know.
I really appreciate your answer. Thank you.
Yes.
Next person, please. Anyone else?
Can I ask a question, Dr. Sati?
Yes, Sofian, you can ask.
Okay, thank you. Doc, my name is Sofian. In one of your slides, you mentioned that inadequate oxidation of NADH to NAD will lead to cellular hypoxia. Does it relate to the uncouplers? In lots of your presentations, you discuss about uncoupling UCP1, UCP3. Does it related to UCP1 or UCP3 in the electron transport chain?
I'm not sure. It's a good question. I would certainly refer you to Dr. Schellenberger's book ozone therapy, and chapters, I think it's 3, 4, and 5, go into a tremendous amount of detail about the ratio of NADH to NAD and how critical that ratio is. And he is the one that I've learned more about uncouplers, but I'm not an expert in uncouplers, so I can't directly answer your question. It's a good question though.
Well, thank you. Thank you.
Dr. Silva, you have a question?
Yes, can you hear me?
Yes, I can.
Yes, Ron, I'd just like to ask you, what is your opinion on this scurvy that's to be divided into the definition of classical scurvy, which we are familiar, and then acute induced scurvy, and chronic scurvy, chronic vagal scurvy. This terminology probably many people understand it. Can you elaborate on it? Thank you.
Yeah, no, I totally am in agreement with you that people, you know, we think, a lot of doctors think of either you have scurvy or you don't, but I think there's all degrees of scurvy. And then Dr. Levy talks about different organs can have different levels of scurvy. And because there may be, the distribution system of the body may not be exactly equivalent, maybe due to blood flow, maybe due to localized oxidative stress will cause the levels of vitamin C to go down in particular organs. And so scurvy is just basically a general term for referring to an inadequate amount of ascorbate in order to quench the degree of free radicals. So again, we're trying to maintain that balance. So it's interesting that we want to have adequate amounts of vitamin C available, but because we don't make it in ourselves, we have to rely upon our daily intake. And so in general, Linus Pauling and basically all the forefathers that have discussed vitamin C, generally it's better to err on the side of taking adequate amounts. I will comment though on, there are people that are concerned that if you take just a small amount of vitamin C, you may be promoting the growth of cancer. And I understand that argument. I think in my mind is that they're probably taking a too small of amount compared to what our ancestors would take. And it probably also depends upon the degree of oxidative stress that particular individual is dealing with. So if you're under a lot of stress, or if you're really wanting to prevent illness, I think you need to err on the side of the Pauling dose, which would be 12 to 18 grams of vitamin C per day. Not everyone needs that, but in the world that we live in, I think there's a greater need for vitamin C than what people think. But I think it does vary with individuals.
Thank you.
Dr. Ron, it's me, Dr. Balchit again. Could somebody actually take too much antioxidants?
Yeah. See, this is, you know, there's one thing I didn't get into is taking a lot of antioxidants have not really proven to be an anti-aging strategy. So really, I think the concept of redox balance is probably more appropriate. So I know that there was a theory that the antioxidant theory of aging has not played out as well as people thought it would. And so we're now moving more into a concept of redox balance. And that may be the topic of a whole nother lecture, because I'm not quite sure how you measure that or how you determine it, but certainly eating, you know, a lot of colorful foods, organic foods, that's a very good thing to do. But just taking a ton of pills may not be the best way to be healthy. So there needs to be some fine distinctions drawn here. But I appreciate your question, because I think redox balance is more important than just taking tons of antioxidants.
Are there any lab tests that one can do, not expensive tests, but simple tests to sort of guide us as to whether somebody's redox balance is off or whether it seems to be reasonable?
Yeah. Well, I don't know for sure. I certainly take, you know, going back to my previous lecture, reverse T3. I'm very intrigued that reverse T3 acts as a kind of a touchstone in terms of helping determine how balanced a person is. Certainly, C-reactive protein, their inflammation levels can be a marker. But I think I really appreciate your question. And I am going to try to pursue that in my own practice. How do we measure redox balance? And Dr. Schallenberger's his bicycle test, where he looks at oxygen utilization, the energy quotient may be a really good way to determine whether or not people are in redox balance.
Anyone else? If there's no more questions. Okay, I think there's no more questions. Dato?
Okay. Thank you very much, Ron, for your wonderful lecture. You enjoyed the lecture as well as I enjoyed the questions.
Yeah, they were good questions.
Yes. Okay, now for the participants, we have an announcement. Once all the recordings are in place, we will send it out to all the participants, and then we will organize an exam with questions as before for module 4, 5, and 6. This will apply for both the certification and the advanced level. So this announcement will be made. We'll get back to you. In the meantime, enjoy your life, be healthy, and I'm very happy that the lectures have turned out to be very useful. I wanted to touch upon mitochondrial health especially as the way forward, and everything links to mitochondrial health, as you have seen in many of these lectures. And we have had wonderful speakers, and we will keep bringing more speakers as well as the good ones we will always retain. And we had very good lectures from Thomas Levy and Dr. Ron, Dr. Patana. And I hope all of you all have enjoyed and feel the same way as I do. Thank you very much. Thank you, Ron.
Thank you.
Thank you.
Thank you, Dr. Ron. Thank you, Dato.
Thank you.
And thank you
Hey,
all participants.
can I get a copy of the recordings of the lectures at some time?
Yes, I will forward to you.
Okay, thank you.
Okay, good night, everyone.
Good night. Good
Okay,
night.
bye-bye. Thank you.
Bye-bye.
Thank you very much.
Bye-bye.
Bye-bye.
Bye.